Hi there, welcome to our IB Biology revision series on section A1.2 nucleic acids. So, the importance of complementary base pairing, DNA replication. This is the copying of information. This can be passed on to daughter cells. Transcription, DNA produces made with one strand from the DNA molecule. Messenger RNA, mRNA, carries the sequence to the ribosome. Translation, the sequence of the base which can be used to determine an amino acid sequence within a polypeptide. mRNA carries codons, which are 3 bases, and then you've got this concept of transfer RNA. This has anticodons that carries 1 amino acid. Ribosomes link the codon to the anticodon and translate it into a specific amino acid in the polypeptide. So, storage of genetic information. DNA stores genetic information in its sequence of nitrogenous bases. Every 3 bases represents a triplet codon. Triplets within the DNA code for 1 of the 20 amino acids. There are 4 different DNA nucleotides that can be arranged as sequence triplets. There are 64 combinations of nucleotides within the triplet coding system. All of those 64 combinations are used in the genetic code, which code 4 amino acids. Genetic uniqueness. It does not matter if the organism is a bacteria, fungus, or an insect. The genetic code is universal. Organisms are different due to the DNA code, from the principle of Darwin and the theory of evolution. So ends of RNA and DNA. The rest of this section is for high-level students only. The ends of DNA and RNA are different. They are referred to 3' and 5'. The 3' end has a pentose or a deoxyribose sugar, which the phosphate group of a nucleotide is linked to this. On a new strand of DNA or RNA, each extra nucleotide is added to the sugar at the 3' end, and this is done by the bonding of a phosphate at the 5' end. Nucleotides are added in a 5 to 3 direction. DNA strands are anti-parallel as they run in the opposite direction, and the diagram of this is represented on the right-hand side. So, purine to pyrimidine bonding. The nitrogenous bases DNA are grouped into pyrimidines and purines. Pyrimidines are smaller as they contain a single ring structure, whereas purines are larger. They contain a double ring structure. A purine is always bonded to a pyrimidine, resulting in 2 strands of DNA being consistent distance from each other. And from an exam tip point of view, adenine and guanine are purines, and pyrimidines are single size ring are thymine and cytosine. So, the Hershey and Chase experiment. So what they wanted to see if genes were DNA or proteins. The T2 virus was used. This was infected with an E. coli, a bacteria. Proteins of T2 were produced inside E. coli, and then when the virus came into contact with this, Hershey and Chase needed to find which part of the virus entered the bacterium. So viruses such as a T2 have DNA and protein, therefore the genes are made of one of these materials. Hershey and Chase prepared 1 strain of T2 and was labeled with radioactive isotope 32P phosphorus and another 35S sulfur. The strains were mixed with E. coli. The mixture was agitated. So interestingly, 32P showed that the viral DNA had been taken up by the bacteria. 32S showed that some protein coats were attached to the bacteria. The experiment showed that the genetic material was composed of DNA rather than protein. So, Shah graphs data. The amount of base is not equal. The percentages were the same amount from different tissues from one species. From other species, the amount differed. DNA is complementary base pairing. Purine bases pair with a pyrimidine. So really important from a nature of science perspective, Shah graph used an identification technique that involved paper chromatography, and basically it was assumed that all 4 bases were of equal percentages. However, Shah graph proved that the content of the bases were not equal, and this disproved the tetranucleotide theory. And for an experiment report, you consider that results agree with the hypothesis and results can disagree with the hypothesis as well, and this has been shown in science many times. Thank you very much for listening to this section on A1.2. We will be adding further revision videos into our IB Biology revision series.